Hello everyone. Audiobook NCRT Class 11th for NEET and CBSE students. Chapter number 8, Cell, the Unit of Life. When you look around, you see both living and non-living things. You must have wondered and asked yourself, what is it that makes an organism living, or what is it that an inanimate thing does not have which a living thing has? The answer to this is the presence of the basic unit of life, the cell, in all living organisms. All living organisms are composed of cells. Some are composed of a single cell and are called unicellular organism, while others like us composed of many cells and are called as multicellular organism. What is a cell? Unicellular organisms are capable of number 1, independent existence, and number 2, performing the essential functions of life. Anything less than a complete structure of a cell does not ensure independent living. Hence, a cell is the fundamental structural and functional unit of all living organisms. Anton von Leeuwenhoek first saw and described a live cell. Robert Brown later discovered the nucleus. The invention of the microscope and its improvement leading to the electron microscope revealed all the structural details of the cell. Cell theory. In 1838, Matthias Schalden, a botanist, examined a large number of plants and observed that all plants are composed of different kind of cells which form the tissue of the plant. At about the same time, Theodor Schwann, a zoologist, in 1839 studied a different type of animal cells and reported that cells had a thin outer layer which is today known as the plasma membrane. Both the scientists were originated from Germany. He also concluded, based on his studies on plant tissues, that the presence of cell wall is a unique character of the plant cells. On the basis of this, Schwann proposed the hypothesis that the bodies of animals and plants are composed of cells and products of cells. Schalden and Schwann together formulated the cell theory. This theory, however, did not explain as to how new cells were formed. Rudolf Virchow, 1855, first explained that cells divided and new cells are formed from pre-existing cells, and this statement is called as omnis cellula e cellula. He modified the hypothesis of the Schalden and Schwann to give the cell theory a final shape. Cell theory as understood today is: number 1, all living organisms are composed of cells and products of cell. Number 2, all cells arise from the pre-existing cell. An overview of a cell. You have earlier observed cells in an onion peel and on human chick cells under the microscope. Let us recollect their structure. The onion cell, which is a typical plant cell, has a distinct cell wall as its outer boundary, and just within it is the cell membrane. The cells of the human cheek have an outside membrane as the delimiting structure of the cell. Inside each cell is a dense membrane-bound structure called a nucleus. This nucleus contains the chromosome, which in turn contain the genetic material DNA. Cells that have membrane-bound nuclei are called eukaryotic, whereas cells that lack a membrane-bound nucleus are prokaryotic. In both prokaryotic and eukaryotic cell, a semi-fluid matrix called cytoplasm occupies the volume of the cell. The cytoplasm is the main arena of the cellular activities in both the plant and animal cells. Various chemical reactions occur into keep the cells in the living state. Beside the nucleus, the eukaryotic cell have other membrane-bound distinct structures called organelles, like the endoplasmic reticulum, which is known as ER, the Golgi complex, lysosome, mitochondria, microbodies, and vacuole. The prokaryotic cell lacks such membrane-bound organelles. Ribosome are the non-membrane-bound organelle found in all cells, both eukaryotic as well as prokaryotic. Within the cell, ribosomes are found not only in the cytoplasm but also within the two organelles, chloroplast, that is in plants, and mitochondria, and on RER, that is rough endoplasmic reticulum. Animal cells contain another non-membrane bound organelles called centrosome, which help in cell division. Cells differ greatly in size, shape, and activities. For example, mycoplasma, the smallest cell, are only 0.3 micrometer in length, while bacteria could be 3 to 5 micrometer. The largest isolated single cell is the egg of an ostrich. Among multicellular organisms, human RBC, that is red blood cells, are about 7 micrometer in diameter. Nerve cells are some of the longest cells. Cells also vary greatly in their shape. They may be disc-shaped, polygonal, columnar, cuboidal, thread-like, or even irregular. The shape of the cell may vary with the function they perform. In figure 8.1, the diagram showing different type of cells. First of all, RBCs whose diameter is around 7 micrometer, which are round and biconcave in shape. White blood cells, which help in providing immunity to the body. They are amoeboid in shape and irregular in shape. Also, they have nucleus. Columnar epithelium cells, which are long and narrow and have nucleus also. Nerve cells, which is the longest or branched cells and have cell body. They have a longest structure, which is axon and dendrite also. A trached, elongated structure present in the xylem of plant. Mesophyll cells, which are round and oval in shape, and they help in photosynthesis because they contain chloroplast. Prokaryotic cell. The prokaryotic cells are represented by bacteria, blue-green algae, mycoplasma, and PPLO, which is pleuropneumonia-like organism. They are generally smaller and multiply more rapidly than the eukaryotic cell. They may vary greatly in shape and size. The four basic shape of bacteria are bacillus, that is rod-like, coccus spherical, vibrio coma shape, and spirillum spiral. The organization of the prokaryotic cell is fundamentally similar, even though prokaryotes exhibit a wide variety of shapes and function. All prokaryotes have a cell wall surrounding the cell membrane except in the mycoplasma. The fluid matrix filling the cell is the cytoplasm. There is no well-defined nucleus. The genetic material is basically naked, not enveloped by a nuclear membrane. In addition to the genomic DNA, the single chromosome circular DNA, many bacteria have small circular DNA outside the genomic DNA. These smaller DNA are called as plasmid. The plasmid DNA confers certain unique phenotypic character to such bacteria. One such character is resistance to antibiotic. In higher classes, you will learn that this plasmid DNA is used to monitor bacterial transformation with foreign DNA. Nuclear membrane is found in eukaryote. No organelles like the ones in eukaryote are found in the prokaryotic cell except for ribosome. Prokaryote have something unique in the form of inclusion. A specialized differentiated form of cell membrane called mesosome is the characteristics of prokaryote. They are essentially enfoldings of the cell membrane. In figure 8.2, diagram showing comparison of eukaryotic cell with other organism. There is a typical eukaryotic cell which is the 10 to 12 micrometer. Typical bacteria is 1 to 2 micrometer. PPLO pleuropneumonia-like organism are about 0.1 micrometer. Viruses are 0.02 to 0.2 micrometer. Cell envelope and its modification. Most prokaryotic cell, particularly the bacterial cell, have a chemically complex cell wall. The cell envelope consists of a tightly bound three-layered structure, that is the outermost glycocalyx, followed by the cell wall and then the plasma membrane. Although each layer of the envelope performs distinct function, they act together as a single protective unit. Bacteria can be classified into two groups on the basis of the differences in their cell envelopes and the manner in which they respond to the staining procedure developed by Gram. With those that take up the Gram stain are called Gram positive, and others that do not are called Gram negative bacteria. Glycocalyx differ in composition and thickness among different bacteria. It could be a loose sheath called the slime layer in some, while in others it may be thick and tough called the capsule. The cell wall determines the shape of the cell and provides a strong structural support to prevent the bacterium from bursting or collapsing. The plasma membrane is selectively permeable in nature and interacts with the outside world. This membrane is similar structurally to that of the eukaryote. A special membranous structure is the mesosome, which is formed by the extension of the plasma membrane into the cell. These extensions in the form of vesicle, tubules or lamellae, they help in cell wall formation, DNA replication and distribution to daughter cell. They also help in respiration, secretion processes to increase the surface area of the plasma membrane and enzymatic content. In some prokaryotes like cyanobacteria, there are other membranous extension into the cytoplasm called chromatophores, which contain pigment. Bacterial cells may be motile or non-motile. If motile, they have thin filamentous extension from their cell wall called flagella. Bacteria show a range in the number and arrangement of flagella. Bacterial flagellum is composed of three parts: filament, hook and basal body. The filament is the longest portion and extends from the cell surface to the outside. Besides flagellae, pili and fimbriae are also surface structures of the bacteria but do not play a role in motility. The pili are elongated tubular structures made up of a special protein. The fimbriae are small bristle-like fibers sprouting out of the cell. In some bacteria, they are known to help attach the bacteria to rocks in streams and also to the host tissues. Ribosome and inclusion bodies. In prokaryote, ribosomes are associated with the plasma membrane of the cell. They are about 15 nanometer by 20 nanometer in size and are made up of two subunits, 50S and 30S units, which when present together form 70S prokaryotic ribosome. Ribosome are the site of protein synthesis. Several ribosomes may attach to a single mRNA and form a chain called polyribosome or polysome. The ribosome of a polysome translate the mRNA into protein. Inclusion bodies. Reserve material in prokaryotic cells are stored in the cytoplasm in the form of inclusion bodies. These are not bound by any membrane system and lie free in the cytoplasm. For example, phosphate granules, synophycean granules and glycogen granules. Gas vacuoles are found in blue, green and purple and green photosynthetic bacteria. Eukaryotic cell. The eukaryotes includes all the protist, plant, animal and fungi. In eukaryotic cell, there is an extensive compartmentalization of cytoplasm through the presence of membrane-bound organelles. Eukaryotic cell possess an organized nucleus with a nuclear envelope. In addition, eukaryotic cell have a variety of complex locomotory and cytoskeleton structures. Their genetic material is organized into chromosomes. All eukaryotic cells are not identical. Plant and animal cells are different as the former possess cell walls, plastid and a large central vacuole which are absent in animal cell. On the other hand, animal cells have centriole which are absent in almost all plant cell. In figure 8.3, these are the diagram which shows number A plant cell and number 2 animal cell. Both are eukaryotic cell. Plant have some additional things. For example, plant have cell wall, but in animals cell wall is absent. Cell wall is a rigid and supportive structure which is absent in animal cell. Let us now look at individual cell organelles to understand their structures and function. Cell membrane. The detailed structure of the membrane was studied only after the advent of the electron microscope in the 1950s. Meanwhile, chemical studies on the cell membrane, especially in the human RBCs, enabled the scientists to deduce the possible structure of plasma membrane. These studies showed that the plasma membrane is mainly composed of lipids and proteins. The major lipids are phospholipids that are arranged in a bilayer. Also, the lipids are arranged within the membrane with the polar head towards the outer side and the hydrophobic tail towards the inner part. This ensures that the non-polar tail of saturated hydrocarbon is protected from the aqueous environment. In addition to phospholipid, membrane also contain cholesterol. Later biochemical investigation clearly revealed that the cell membranes also possess protein and carbohydrate. The ratio of protein and lipid varies considerably in different cell types. In human beings, the membrane of the erythrocyte has approximately 52% protein and 40% lipid. Depending on the ease of extraction, ease of extraction, membrane proteins can be classified as integral and peripheral. Peripheral proteins lie on the surface of the membrane, while the integral proteins are partially or totally buried in the cell membrane. In figure 8.4, fluid mosaic model of the plasma membrane is shown, in which there is a blue outer line and blue lower line. Blue outer and lower line is made up of basically lipid bilayer, and it is mainly phospholipid bilayer, in which the spherical portion represent the head and there is a tail portion also, and both are made up of lipid. There is peripheral protein and coated with the sugar, that is glycosylation, and there is integral protein or peripheral or extrinsic protein also. Cholesterol molecules are also shown in the figure. An improved model of the structure of cell membrane was proposed by Singer and Nicholson in 1972, widely accepted as fluid mosaic model. According to this, the quasi-fluid nature of the lipid enable lateral movement of the protein within the overall bilayer. This ability to move within the membrane is measured as its fluidity. The fluid nature of the membrane is also important for the point of view of functions like cell growth, formation of intercellular junctions, secretions, endocytosis, cell division, etc. One of the most important functions of the plasma membrane is the transport of the molecules across it. The membrane is selectively permeable to some molecules present on either side of it. Many molecules can move briefly across the membrane without any requirement of energy, and this is called the passive transport. Neutral solutes may move across the membrane by the process of simple diffusion along the concentration gradient, that is from higher concentration to the lower. Water may also move across this membrane from higher to lower concentration. Movement of water by diffusion is called osmosis. As a polar molecule cannot pass through the non-polar lipid bilayer, they require a carrier protein of the membrane to facilitate their transport across the membrane. A few ions or molecules are transported across the membrane against their concentration gradient, that is from lower to the higher concentration. Such a transport is an energy dependent process in which ATP is utilized and is called active transport. For example, sodium potassium pump. Cell wall. As you may recall, a non-living rigid structure called the cell wall forms an outer covering for the plasma membrane of fungi and plants. Cell wall not only gives a shape to the cell and protects the cell from mechanical damage and infection, it also helps in cell-to-cell interactions and provide barrier to undesirable macromolecule. Algae have cell wall made of cellulose, galactans, mannans and minerals like calcium carbonate, while in other plants it consists of cellulose, hemicellulose and pectins and proteins. The cell wall of a young plant cell, the primary cell wall, is capable of growth, which gradually diminishes as the cell matures, and the secondary wall is formed on the inner towards membrane side of the cell. The middle lamella is a layer mainly of calcium pectate which holds or glues the different neighboring cells together. The cell wall and middle lamellae may be traversed by plasmodesmatal connections which connect the cytoplasm of the neighboring cell. Endomembrane system. While each of the membranous organelle is distinct in terms of its structure and function, many of these are considered together as an endomembrane system because of their functions are coordinated. The endomembrane system include endoplasmic reticulum, Golgi complex, lysosome, vacuoles. Since the function of the mitochondria, chloroplast and peroxisomes are not coordinated with the above component, these are not considered as a part of the endomembrane system. Endoplasmic reticulum. Electron microscopic studies of eukaryotic cell reveal the presence of a network or reticulum of tiny tubular structures scattered in the cytoplasm that is called the endoplasmic reticulum. Hence, ER divides the intracellular space into two distinct compartments, that is luminal inside ER and extraluminal cytoplasm compartment. The ER often shows ribosomes attached to the outer surface. The endoplasmic reticulum bearing ribosomes on their surface is called rough endoplasmic reticulum. In the absence of ribosome, they appear smooth and are called smooth endoplasmic reticulum, that is SER. RER is frequently observed in the cells actively involved in protein synthesis and secretion. They are extensive and continuous with the outer membrane of the nucleus. The smooth endoplasmic reticulum is a major site for synthesis of lipid. In animal cell, lipid-like steroidal hormones are synthesized in SER. Golgi apparatus. Camillo Golgi in 1898 first observed densely stained reticular structures near the nucleus. These were later named as Golgi bodies after him. They consist of many flat disc-shaped sac or cisternae of 0.5 micrometer to 1 micrometer in diameter. These are stacked parallel to each other. Varied number of cisternae are present in a Golgi complex. The Golgi cisternae are concentrically arranged near the nucleus with distinct convex cis or the forming phase or concave trans or the maturing phase. The cis and the trans phase of the organelle are entirely different but interconnected. The Golgi apparatus principally performs the function of packaging materials to be delivered either to the intracellular targets or secreted outside the cell. Material to be packaged in the form of vesicle from the ER fuse with the cis phase of the Golgi apparatus and move towards the maturing phase. This explains why the Golgi apparatus remains in the close association with the endoplasmic reticulum. A number of proteins synthesized by the ribosome on the endoplasmic reticulum are modified in the cisternae of the Golgi apparatus before they are released from its trans phase. Golgi apparatus is an important site of formation of glycoprotein and glycolipid. Lysosome. These are membrane-bound vesicular structure formed by the process of packaging in the Golgi apparatus. The isolated lysosomal vacuoles have been found to be very rich in almost all type of hydrolytic enzyme, that is hydrolases, lipases, proteases, carbohydrases, optimally active at the acidic pH. These enzymes are capable of digesting carbohydrate, proteins, lipids and nucleic acid. Vacuole. The vacuole is a membrane-bound space found in the cytoplasm. It contains water, sap, excretory product and other materials not useful for the cell. The vacuole is bound by a single membrane called tonoplast. In plant cell, the vacuole can occupy up to 90% of the cell volume of the cell. In plants, the tonoplast facilitates the transport of a number of ions and other materials against concentration gradient into the vacuole. Hence, their concentration is significantly higher in the vacuole than in the cytoplasm. In amoeba, the contractile vacuole is important for osmoregulation and excretion. In many cells, as in protists, food vacuoles are formed by engulfing the food particle. Mitochondria. Before that, in figure 8.5, endoplasmic reticulum are shown and RER is shown and SER is also shown. Ribosome present on the cisternae and that is known as the rough endoplasmic reticulum, and absence of ribosome over the cisternae tubule are considered as a smooth endoplasmic reticulum, and ER are the extension from the outer nuclear membrane, and nucleus have two membrane, inner membrane and outer membrane, and there is a perinuclear space in between them in which nuclear pores are present. Golgi apparatus also have stack-like structures, cisternae, they are called as a cisternae, and it has a forming phase which is convex phase, also known as the cis phase, and the opposite end of the cis phase is known as the trans phase or the maturing phase or concave phase. Now mitochondria. Mitochondria, singular is considered as mitochondrion, unless specifically stained, are not easily visible under the microscope. The number of mitochondria per cell is variable depending on the physiological activity of the cell. In terms of the shape and size also, considerable degree of variability is observed. Typically, it is sausage shape or cylindrical, having a diameter of 0.2 to 1 micrometer, average is 0.5 micrometer and the length is 1.0 to 4.1 micrometer. Each mitochondrion is a double membrane bound structure with the outer membrane and the inner membrane dividing its lumen distinctly into two aqueous compartments. That is the outer compartment and the inner compartment. The inner compartment is filled with a dense homogeneous substance called the matrix. The outer membrane forms a continuous limiting boundary of their organelles. The inner membrane forms a number of enfoldings called the cristae, and singular is considered as crista. Towards the matrix, the cristae increases the surface area. The two membranes have their own specific enzyme associated with the mitochondrial function. Mitochondria are the site of aerobic respiration. They produce cellular energy in the form of ATP, hence they are called powerhouses of the cell. The matrix also possesses a single circular DNA molecule. The outer membrane forms a continuous limiting boundary of the organelle. The inner membrane forms a number of enfoldings called cristae towards the matrix. The cristae increases the surface area. The two membranes have their own specific enzyme associated with the mitochondrial function. Mitochondria are the sites of aerobic respiration. They produce cellular energy in the form of ATP. Hence, they are called as powerhouses of the cell. The matrix also possesses a single circular DNA molecule, a few RNA molecule, ribosome 70S, and the components required for the synthesis of protein. The mitochondria divide by fission. In structure 8.7, there is outer membrane, there is inner membrane, there is a matrix, intermembrane space also there, and crista, which is the enfolding of the inner membrane, are also shown. Plastid. Plastids are found in all plant cells and in euglenoids. These are easily observed under the microscope as they are large. They bear some specific pigments, thus imparting specific colors to the plant. Based on the type of pigment, plastids can be classified into chloroplast, chromoplast, and leucoplast. The chloroplast contain chlorophyll and carotenoids pigments, which are responsible for trapping light energy essential for photosynthesis. In the chromoplast, fat-soluble carotenoid pigments like carotene, xanthophyll, and others are present. This gives the part of the plant a yellow, orange, or red color. The leucoplasts are the colorless plastids of varied shapes and size with stored nutrients. A myeloplast store carbohydrate starch, for example potato. Elioplast stores oils and fats, whereas the aleuroplast store proteins. Majority of the chloroplasts of the green plants are found in the mesophyll cells of the leaf. These are lens-shaped, oval, spherical, discoid, or even ribbon-like organelles having variable length 5 to 10 micrometer and width 2 to 4 micrometer. Their number varies from 1 per cell of the Chlamydomonas, a green alga, to 20 to 40 per cell in the mesophyll. Like mitochondria, the chloroplasts are also double membrane bound. One of the two inner chloroplast membrane is relatively less permeable. The space limited by the inner membrane of the chloroplast is called the stroma. A number of organized flattened membranous sacs called the thylakoids are present in the stroma. Thylakoids are arranged in stacks like the piles of coins called grana, singular granum, or the intergranal thylakoids. In addition, there are flat membranous tubules called the stroma lamellae connecting the thylakoid of the different grana. The membrane of the thylakoid encloses a space called a lumen. The stroma of the chloroplast contain enzyme required for the synthesis of carbohydrate and proteins. It also contains small double-stranded circular DNA molecules and ribosomes. Chlorophyll pigments are present in the thylakoids. The ribosomes of the chloroplast are smaller 70S than the cytoplasmic ribosomes 80S. Outer membrane, inner membrane, granum, thylakoids, stroma lamellae, and stroma in figure 8.8, that is a sectional view of the chloroplast. 8.5.6 Ribosome. Ribosomes are the granular structure first observed under the electron microscope as a dense particle by the George Palade in 1953. They are composed of ribonucleic acid. RNA and proteins are not surrounded by any membrane. The eukaryotic ribosome are 80S while the prokaryotic ribosome are 70S. Each ribosome has two subunits, large and smaller subunit. The two subunits of 80S ribosome are 60S and 40S, while that of 70S ribosomes are 50S and 30S. Here S Swedberg unit stands for the sedimentation coefficient. It is indirectly a measure of the density and size. Both 70S and 80S ribosomes are composed of two subunits. Cytoskeleton. An elaborate network of the filamentous proteinaceous structures consisting of microtubules, microfilament, and intermediate filaments present in the cytoplasm is collectively referred to as a cytoskeleton. The cytoskeleton in a cell are involved in many functions such as mechanical support, motility, maintenance of the shape of the cell. Cilia and flagella. Cilia singular cilium and flagella singular flagellum are hair-like outgrowths of the cell membrane. Cilia are the small structures which works like oar, causing the movement of either the cell or the surrounding fluid. Flagella are comparatively longer and responsible for cell movement. The prokaryotic bacteria also possess flagella, but these are structurally different from that of the eukaryotic flagella. The electron microscopic study of a cilium or a flagellum show that they are covered with plasma membrane. Their core, called axoneme, possesses a number of microtubules running parallel to the long axis. The axoneme usually has 9 doublets of radially arranged peripheral microtubules and a pair of centrally located microtubules. Such an arrangement of the axonemal microtubule is referred to as 9+2 array. The central tubules are connected by a bridge and is also enclosed by a central sheath, which is connected to one of the tubules of each peripheral doublets by a radial spoke. Thus, there are 9 radial spokes. The peripheral doublets are also interconnected by linkers. Both the cilium and flagellum emerge from centriole-like structure called the basal body. In the figure 8.10, section of cilia and flagella showing different parts. A is electron micrograph, B is diagrammatic representation of the internal structure. In the B figure, there is a plasma membrane, and inside the plasma membrane there is a doublet of the microtubule which are arranged peripherally, and in between the peripheral microtubules there is a interdoublet bridge, that is a bridge between the two microtubules, and there is a central microtubule under the central sheath which are 2 in number, and from the peripheral microtubule there is origination of the radial spoke towards the central sheath. Now, centrosome and centriole. Centrosome is an organelle usually containing two cylindrical structures are called centriole. They are surrounded by amorphous pericentriolar materials. Both the centrioles in the centrosome lies perpendicular to each other, in which each has an organization like the cartwheel. They were made up of 9 evenly spaced peripheral fibrils of tubulin protein. Each of the peripheral fibril is a triplet. The adjacent triplets are also linked. The central part of the proximal region of the centriole is also proteinaceous and called the hub, which is connected by the tubules of the peripheral triplets by a radial spoke made up of protein. The centrioles form the basal body of cilia or flagella, the spindle fiber that give rise to spindle apparatus during cell division in animal cell. Nucleus. Nucleus as an organelle, as a cell organelle, was first described by Robert Brown as early as 1831. Later, the material of the nucleus stained by the basic dye was given the name chromatin by Fleming. The interphase nucleus, that is nucleus of a cell when it is not dividing, has a highly extended and elaborate nucleoprotein fibers called chromatin. Nuclear matrix and one of the more spherical bodies called nucleoli. Electron microscopy has revealed that the nuclear envelope, which consists of two parallel membranes with a space between 10 to 15 nanometer called the perinuclear space, forms a barrier between the materials present inside the nucleus and that of the cytoplasm. The outer membrane usually remains continuous with the endoplasmic reticulum and also bear ribosome on it. At a number of places, the nuclear envelope is interrupted by minute pores which are formed by the fusion of its two membranes. These nuclear pores are passes through which movement of RNA and protein molecule takes place in both the directions between the nucleus and the cytoplasm. Normally, there is only one nucleus per cell. Variations in the number of nuclei are also frequently observed. Can you recollect a name of organism that have more than one nucleus per cell? For example, in fungus, there is Rhizopus, a fungus, and it has aseptate mycelium, aseptate hyphae. So there is a multinucleated condition which is known as a cynocytic condition also. Some mature cells even lack of nucleus, for example erythrocytes of many mammals and sieve tube cells of vascular plant. Would you consider these cells as living? Yes, we consider them as a living cells because sieve cells are controlled by the companion cell nucleus and RBCs shows anaerobic respiration. So these are the cells which have plasma membrane, that's why they are considered as living. The nuclear matrix or the nucleoplasm contains nucleolus and the chromatin. The nucleoli are spherical structures present in the nucleoplasm. The content of nucleolus is continuous with the rest of the nucleoplasm as it is not a membrane-bound structure. It is a site for the active ribosomal RNA synthesis. Larger and more numerous nucleoli are present in the cells actively carrying out protein synthesis. So, structure of nucleus, nucleoplasm, nucleolus, nuclear pore, and nuclear membrane. You may recall that the interface nucleus has a loose and an indistinct network of nucleoprotein fibers called chromatin. But during different stages of cell division, cell shows a structured chromosome in the place of nucleus. Chromatin contain DNA and some basic protein called histone, some non-histone proteins and also RNA. A single human cell has approximately 2-meter long thread of DNA distributed among its 23 pairs chromosome. You will study the details of DNA packaging in the form of chromosome in class 12th. Every chromosome visible only in dividing cell essentially has a primary constrictions or the centromere on the sides of which disc-shaped structures called neutocores are present. Centromere holds two chromatid of a chromosome. Based on the position of the centromere, the chromosome can be classified into 4 types. The metacentric chromosome has a middle centromere forming 2 equal arms of the chromosome. The submetacentric chromosome has a centromere slightly away from the middle of the chromosome, resulting into one shorter arm and a one longer arm. And in case of acrocentric chromosome, the centromere is situated close to its one end, forming one extremely short and one very long arm, whereas the telocentric chromosome has a terminal centromere. So, types of chromosome based on the position of centromere are shown in the figure, in which first is metacentric, second one is a submetacentric, third one is acrocentric, and the fourth one is a telocentric. Short arm is considered as a P arm and long arm is considered as P arm, and short arm is considered as short arm is P arm and long arm is Q arm. There is also secondary constrictions which has satellite and nucleolar organizing region, that is the NOR region. And in the middle there is a constricted area that is considered as a centromere. Around the centromere, there is a disc-shaped proteinaceous structure which is considered as the neetochore, and spindle fiber gets attached with the neetochore. Sometimes a few chromosomes have non-staining secondary constrictions at a constant location. These gives the appearance of a small fragment called satellite. Microbodies. Many membrane-bound minute vesicles called microbodies that contain various enzymes are present in both plant and animal cell. Summary. All organisms are made up of cells or aggregates of cells. Cells vary in their shape, size, and activities functions. Based on the presence or absence of a membrane-bound nucleus and other organelles, cells and hence organism can be named as eukaryotic or prokaryotic. A typical eukaryotic cell consists of a cell membrane, nucleus, and cytoplasm. Plant cells have a cell wall outside the cell membrane. The plasma membrane is selectively permeable and facilitates transports of several molecules. The endomembrane system includes Golgi complex, lysosome, and vacuoles. All the cell organelles perform different but specific functions. Centrosome and centriole form the basal body of cilia and flagella that facilitate locomotion. In animal cell, centrioles also form spindle apparatus during cell division. Nucleus contains nucleoli and chromatin network. It not only controls the activities of organelles but also plays a major role in heredity. Endoplasmic reticulum contains tubules or cisternae. They are of two types, rough and smooth. Help in transport of substances, synthesis of proteins, lipoproteins and glycogen. The Golgi body is a membranous organelle composed of flattened sac. The secretion of the cells are packed in them and transported from the cell. Lysosomes are single membranous structures containing enzyme for digestion of all type of macromolecules. Ribosomes are involved in protein synthesis. These occur freely in the cytoplasm or are associated with Mitochondria help in oxidative phosphorylation and generation of adenosine triphosphate. They are bound by double membrane. The outer membrane is smooth and inner one folds into several cristae. Plastids are pigment containing organelles found in the plant cells only. In plant cells, chloroplasts are responsible for trapping light energy essential for the photosynthesis. The grana in the plastid is the site of light reaction and the stroma of the dark reaction. The green colored plastids are chloroplasts which contain chlorophyll, whereas the other colored plastids are chromoplasts, which may contain pigments like carotene and xanthophyll. The nucleus is enclosed by nuclear envelope, a double membrane structure with a nuclear pore. The inner membrane encloses a nucleoplasm and chromatin material. Thus, cell is the structural and functional unit of life. Exercise. Thank you.